Hello, my name is Joshua Mugabe. I welcome you to Chromosomes and Fertilization. Fertilization occurs when the nucleus of a male reproductive cell combines with the nucleus of the female reproductive cell. So if we look here, this is the ovum. This is the reproductive cell of the female. And here we have the sperms. And these are the male reproductive cells. So when they come towards the female reproductive cell, it is only one that will penetrate through the cell wall or cell membrane of the ovum, and it is the one whose nucleus will combine with the nucleus of the female reproductive cell, and then fertilization would have occurred. The reproductive cells are called gametes. So these ones that we have, these are gametes. Those are the sperms. They are the male reproductive cells. We call them the gametes. And then this one that we have here, this one is the ovum, and it is the female reproductive cell, or what we call a female gamete. In animals, the male gamete is the sperm cell, and the female gamete is the ovum. Like we have already seen, the sperms, these are the male gametes, that is in animals, and the ovum is the female gamete. Many thousands of sperm cells, they swim towards the ovum, but only one of them will penetrate and fertilize it. Like the way we are seeing here, many of them will swim towards the ovum, but it is going to be only one. This sperm will fertilize the ovum. It is only this one. As soon as it has entered, the mechanism is going to be created, which is going to prevent the entry of the other sperms into the ovum. So it is only this sperm which will penetrate and fertilize the ovum. In the vertical section through the ovary of the flowering plant, the pollen grain lands on the stigma. Then they produce the pollen tubes, which grow through the ovary to reach the ovule. So when you look here, you will see on top the pollen grains that have landed on the stigma. And when they land on the stigma, now they produce the pollen tubes, and these pollen tubes will grow going towards the ovary. This is our ovary, which has been cut through vertical section so that we can see the ovules inside. So the pollen tubes will grow and move towards the ovule. This is our ovule here, and inside we have the egg cell, and then the pollen tube has brought the male gamete. The male gamete is a cell in the pollen grain, and the female gamete is an egg cell in the ovule. So the male gamete is coming with the pollen tube. The pollen tube is growing from the pollen grain, bringing the male gamete, and it is bringing it to the ovule, which contains the female gamete, which is the egg cell. So when it reaches here, the nucleus of the male gamete will enter into the egg cell, and then fertilization or pollination will take place. So the pollen cell reaches the egg cell through the pollen tube. So it reaches the egg cell through the pollen tube. When the male and the female gametes combine, resulting cell is called a zygote. So here when we are saying, for example, in plants, we have a pollen nucleus which is moving towards the egg cell, and when the two of them combine together, now we form a zygote. This product that we have got is what we call a zygote. The same applies in animals, and in animals, when the sperm reaches the ovum, it is going to combine with the ovum, and when the two combine together, now they form a product, and this product that they are formed is what we are calling a zygote. So the combination of a male and a female gamete results into a cell, a full-fledged cell, which we call a zygote. The result of fertilization, the chromosomes from the male and the female cells, they are combined in the same nuclear membrane. So when the male and the female combine, they form a zygote. Now the nucleus of the pollen and the nucleus of the egg cell, they brought chromosomes, and when they come together, those chromosomes are going to combine together in the nuclear membrane. So here in plants, we shall now have cell division, which will take place. The cell that has been formed, it will undergo cell division by mitosis, increasing in the number, and later we shall form an embryo, which is going to form the plumule and the radicle. And the same also applies to the sperms. When they come and mix with the ovum, fertilization takes place. The sperm came with the chromosomes and the ovum came with the chromosomes. They combine together, and when they have combined together, we form a zygote. Now the zygote will start dividing. That cell is going to start dividing, forming 2 cells, forming 4, forming 8, forming many cells. That is cell division by mitosis, and then we shall form an embryo. Do you see a problem with this? Do you see a problem with this? The problem, do you see any problem of a pollen's nucleus combining with the egg cell? Do you see any problem with a sperm combining with an ovum? We are going to get a cell. We are getting a cell. Do you see any problem with that? You can pause the video and try to think about the problem. Identify it, then you can continue. The problem is that the full complement of the male and the female chromosome combine, the zygote and the embryo would have twice as many chromosomes as its parent. So the problem here we are having, if we are saying, for example, in animals, a full animal has got 46 chromosomes. So if we have 46 chromosomes, and then also the ovum comes with 46 chromosomes, which means that the number of the zygote or the embryo is going to double the number of chromosomes of the parents. That is a problem which we are going to see. If 46 male chromosomes combined with 46 female chromosomes, the offspring would have 92 chromosomes in their cells. So that is now the problem which we are likely to get. If we have the sperm which are coming from the male and the ovum which are coming from the female, if this one has got 43, 46, and this one also has got 46, if they come, then the cell that we are going to form, or the zygote or the embryo of the offspring, will have 92 chromosomes in each cell. That is going to be the problem. And the next generation would have 184. So if the offspring that the other one we formed had now 92, that means that now if it also forms another offspring, that generation will have 184 chromosomes. So that means that the generations that are proceeding, the number of chromosomes in the cell will go on increasing and increasing. So that one would be a problem. In fact, when the gametes are formed, the number of chromosomes is halved, so that the zygote ends up with the same number of chromosomes as its parent. So before fertilization takes place, the number of chromosomes in the gametes is halved. For example, we have the sperm. Here the number is going to be halved, and here the number will be halved. If the normal animal has got, a normal animal cell has got 46 chromosomes, therefore that number is going to be halved. When the sperms are formed, they will have 23 chromosomes, and even the ovum will have 23 chromosomes. So that when fertilization takes place, 23 and 23 will combine, and the cell will have now 46 chromosomes, which is the same number as the parent number of chromosomes. So during gamete formation, the number of chromosomes is halved in the gametes. The type of cell division which gives rise to gametes is called meiosis. So when the gametes are going to be formed, there is cell division which takes place, and that type of cell division is called meiosis. And meiosis is the one that ends up dividing or having the number of chromosomes in the gametes. At cell division, before the chromatids separate, the chromosomes are shared equally between the two daughter cells. So now we are having our cell is going to undergo cell division. The cells are going to undergo cell division. Now, what is going to happen first before cell division takes place? The chromosomes, we are going to have chromatids, and these chromatids are going to separate, and they will be shared equally between the two daughter cells that are going to be formed. The chromosomes appear shorten and thicken as in mitosis. So now here we are going to form the gametes. And the first stage, when we are undergoing meiosis, the chromosomes are going to become short. And when they become short, they will become thick. And when they become short and thick in the nucleus, now they will start to become visible. We shall be able to see them in the nucleus because they have become short and they have thickened. The blue chromosomes are from the male parent, and the red chromosomes, they are from the female parent. Now we are taking this cell that it is going to undergo cell division. Now we know that if it is an animal cell, we are likely to have 23 pairs of chromosomes. That makes it 46 chromosomes. But here when you look at our cell, we have only 4 chromosomes, which means they are only 2 pairs of chromosomes. Because the 46 chromosomes, they are very many to be fixed in a small area, we are going only to use 4 chromosomes only. And those 4 chromosomes, those are the 2 pairs. Instead of using the 23 pairs of chromosomes, we are going to use only 2 pairs of chromosomes so that we can easily understand and follow the process of meiosis. So now when we have picked 2 pairs, that means that every pair, for example, this long one and this long one, that is 1 pair, and this short one and this short one, it is also the second pair. But of this long one, the 2, one of them, which is the blue one, this one came from the parent cell, which is the sperm, and this one came from the female parent, which is the ovum. So the 2 will pair up and they form a pair. So that's why we are saying that the blue came from the male parent and the red came from the female parent. And when you look at these short ones also, this is also a pair. But one of the chromosome, this blue one, came from the sperm, which is the male gamete, and this one came from the female gamete, which is the ovum. And when fertilization took place, there was combination or combining or bringing all these chromosomes together. So we have 2 pairs that we are going to use. The blue are the ones that came from the male parent, and the red are the ones that came from the female parent. So we are going to undergo meiosis and we are going to see what is going to take place in the cell. Now, the 2 long chromosomes and the 2 short chromosomes are called homologous pairs. So we have seen that this blue one, which is long, and this blue one, which is also this red one, which is long, they come and combine together. They are always moving together. Therefore, we call them a pair of chromosomes. And when they are moving together, now that pair is called a homologous chromosome or a homologous pair. Now, this short one, that one came from the male parent gamete, and this one came from the female parent gamete. The two will move together, and when they are moving together as a pair, we call them a homologous pair. So the chromosomes have already replicated, but the chromatids are not obvious at this stage. Now, replication, it's whereby this chromosome here, which is blue, and this chromosome here, which is red, each of the chromosome is going to replicate. Replicating means it is making a copy of itself. This one will make a copy of itself, and this one will make a copy of itself. And they will stay together as one chromosome here, but they are two copies. Now those copies, we shall call them chromatid. Now this chromosome will have made a copy of itself, so we shall call them now chromatids. Excuse me. And this one will make a pair of itself, and then we shall call them chromatids. So this chromosome will have two chromatids, and this one will also have two chromatids. The homologous chromosomes come and lie close alongside each other and behave like a single chromosome. So we have now the homologous chromosome. This one that came from the female parent will come and align with this one that came from the male parent, and they will lie along closely with each other, and now we call them the homologous chromosome. The two of them moving together is what we are calling the homologous chromosome, moving as if it is a single chromosome. Then even these ones will lie alongside each other, and we shall refer to them as homologous chromosome. The nuclear membrane will disappear. Now we are starting the process of division. What is going to happen? The membrane which was covering the chromosomes inside is going to disappear. That membrane will disappear. That is the first part. And then the spindles are going to form. We are going to form the spindle fibers. Spindle fibers, they are these thread-like structures. These thread-like structures are the ones that we are calling the spindles. These ones are the ones that we call the spindles. They will form. And after the spindles have been formed, now at this stage, it is the chromosomes, not the chromatids, which are going to separate. So we had the homologous chromosomes. They were lying together. This one was closely together with this chromosome. Now they will separate. This one will go this side, and the other one, the other chromosome, will go the other side. And even these two long chromosomes that were lying close to each other, which we were calling the homologous chromosome, they are going to separate. Now this one will move up, and this one will go down. Now the cell will start to constrict. Now we are going to see the cell. The whole of this one has been one cell. Now it will start to constrict in the middle, becoming smaller or shorter in the middle. It is starting to constrict in the middle, meaning that it is dividing itself in the middle. Now, one long chromosome and one short chromosome, they go to either end of the dividing cell. So here we are seeing that one long chromosome has gone to one side, and one long chromosome of the other side also goes to the other side. So the cells now contain only 2 rather than 4 chromosomes. So now here we are having our cell, as we are seeing the cell excuse me, is dividing in the middle. As it is dividing in the middle, we are going to end up having 2 cells. One cell up and another cell down. But you will see that the cell up is having now only 2 chromosomes, and the cell down is also having only 2 chromosomes. By this time, the chromatids have become clear. Now you can see that each chromosome now has the paired chromatids. Now the chromatids are visible, are seen. You can see that this chromosome has got the chromatids and this one has got the chromatid. 2 new spindles form at right angles to the original spindle. So previously we had one cell, as you remember, and it had spindles forming from that direction, up, down. Now we have got 2 cells that have been formed, and one chromosome, 2 chromosomes went up, 2 chromosomes went up, and 2 chromosomes went down. But we ended up seeing that each chromosome which had gone up, we could see the sister chromatid. It had already replicated with the sister chromatid. 1 chromosome with 2 chromatids. And this one was also 1 chromosome with 2 chromatids. Now here we are seeing that now the new spindles are forming, and these new spindles are forming at a right angle to the original spindles. These ones that we are seeing were the original spindles, these ones going up and down. Now we are forming new spindles. These are the new spindles, these ones that you are seeing. These new spindles that are forming are at a right angle to the original where these ones which were coming from up and down. So the new ones that are forming are the ones that are at a right angle to the new spindles, to the original spindles. At this stage, the chromatids which separate and pass to opposite end of the cell. So we saw originally that the cell had divided itself into 2 to form the upper cell and the lower cell. The upper cell we had 2 chromosomes, and the lower cell we had 2 chromosomes. But the chromosomes had already replicated and formed sister chromatids. Now after forming now these new spindles, now it is the sister chromatids that are going to separate. So you will see the chromosome will separate and the sister chromatids will be going to opposite poles, as you are seeing here, this one was one chromosome, but the chromatids are now separating. This chromatid goes this side and this chromatid goes this side. So the same applies to the lower cell. You would see that this was one chromosome, all of it with sister chromatids, but now you're seeing the sister chromatids going to opposite poles. So it's what we are saying at this stage. It is the chromatids which separates. The chromatids are the ones separating, and they pass to opposite ends of the cells. So they are moving to opposite ends. This one is going this side, this chromatid is going this side, and this chromatid is going this side, this one is going to the other side of the cell. The same applies to the upper cell. This chromatid is going this side, and this chromatid is going this side, this one is going this side, and this one is going the other side of the cell. The cell constricts in the plane at right angles to the first constriction. So now, our first constriction, we still remember our first constriction of the first cell was in the middle, which is this constriction here. Now, after the chromatids have moved to opposite poles, we are now seeing the cell constricting in the plane at a right angle to the first constriction. This was the first constriction. Now we are seeing the second constriction coming at the right angle to the original constriction. We are getting now a second constriction which is at the right angle to the original constriction. Now the cell division is completed, forming 4 gametes, each with half the number of chromosomes of the parent cell. So because of the different constrictions, we had the original constriction was here to form 2 cells. Now again we had another constriction in here. Now they have formed 4 cells. Now this one is having half the number of chromosome of the parent cell. So this one is having 2 chromosomes. 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 Now these are 4 cells. But remember, we originally had 1 cell, and that 1 cell had 4 chromosomes. But now we are having 4 cells, and each cell is having 2 chromosomes, which is half the number of chromosomes of the parent cell. So that type of division or cell division is what we call meiosis, whereby the cell divides and it ends up forming 4 daughter cells, but each cell is having half the number of chromosomes to that of the parent cell. The parent cell we started with had 4 chromosomes, and at the end of the cell division we are having 4 cells, and these cells, each cell is having half the number of chromosomes of the parent, which are 2 chromosomes. In human body cells, they contain 46 chromosomes. That is a cell. But the gametes contain only 23 chromosomes. If you look here, this is a sperm mother cell. Sperm mother cell, it has 46 chromosomes. This is the cell. But when it undergoes cell division, it forms 4 sperms, which are the daughter cells. And these ones, each one is having 23, 23, 23, 23. So the sperms are produced by meiosis. So the human body cell contains 46 chromosomes. But when it undergoes meiosis, to form the gametes end up with only 23 chromosomes, as we are seeing here. Excuse me. So here the sperm mother cell has got 46 chromosomes. But when it undergoes meiosis cell division, it ends up forming 4 daughter cells. And these ones, each one has got 23. Now these ones are the ones that we are calling the sperms, or now what we call the gamete cells. Now when you go to the ovum mother cell, it has got 46 chromosomes. So it divides and it produces 4 daughter cells. Now they overproduced by meiosis, but only one of them develops to maturity. So the other ones will degenerate, and it will be only one which is going to develop, and you will see it will be having 23 chromosomes. So it is this one with the 23 which will combine with this sperm, which also has 23, and after fertilization they are going to form a zygote which is going to have 46, like the original parent cell. So after fertilization, the number is restored to 46. So we are seeing that this is the parent and this is the parent who are having 46. The parent cells are having 46. So when they produce the gametes are going to have half the number of the parent, which is 23. So they will produce half the number of the parent. Now these ones are our gametes. Now these gametes, when they come together, they are going now to combine. And when they combine, then now the 23 and the 23 are going to come together, and we are going to get a zygote which has got now 46. It has now 46 number of chromosomes, which are the same number of chromosomes like the parent cell. So after fertilization, the number is going to be restored back to 46. In humans and most of the mammals, the ovum mother cell produces 4 cells by meiosis, but only one of these goes and becomes the gamete. So we have seen here the ovum mother cell is going to produce 4 cells, and these 4 cells that are produced, it is going to be only one of them which is going to form or to become the gamete. So the ovum produced by meiosis, but only one develops to maturity. The number of chromosomes in the body cell is called diploid number. So here in the body cell we have 46 chromosomes. This 46 chromosome is what we are calling a diploid number. Then the number of chromosomes in the gametes is called haploid number, which is half the diploid number. So when we form the gametes here, you will see that for them they are having half the number of the parent cell. So for these ones, we are going to call them haploid. Haploid meaning they are a half the parent number of chromosomes. The diploid number in humans is 46. So the diploid number of chromosome in the human cells is 46 chromosome. Then the haploid number in the gamete is 23, meaning that the gametes are having 23 chromosomes. Therefore, they are half the number of the parent cells. Cell division continues by mitosis, so all cells will contain 46 chromosomes. So after forming our zygote, after fertilization, remember the sperm brought 23 and the ovum brought 23, and when they combined they formed 46. And after forming 46, now this 46 cell is going to undergo cell division, and that cell division it is going to undergo is by mitosis. Mitosis will end up producing 2 daughter cells, but these 2 daughter cells each will have the same number of chromosomes like the parent cell. So this one was 1 cell, it will divide and they will become 2 cells, but those 2 cells are going to have the same number of chromosomes. Then they will also go on dividing and dividing and dividing, that is during the array embryo stage. And every cell that is formed by the cell division of mitosis, and each cell is going to be having 46 number of chromosomes. The genes for any one characteristics occupy a corresponding position on the homologous chromosome. Now, previously we have talked about the homologous chromosomes. We say the homologous chromosomes, those are chromosomes that come and align themselves together and they move together. Therefore, we refer to them as homologous chromosomes. Now, these ones are homologous chromosomes. And these ones too will align themselves together. We shall call them homologous chromosomes. And also, if you recall, we say that if we take the blue chromosomes to have come from the father, the red chromosomes, they came from the mother. So when they align together, they become what we call the homologous chromosomes. Now, these homologous chromosomes, they have got what we call genes. The genes are located on the chromosomes. Now, those genes, they control particular characteristics. Let us look at this homologous chromosome here. If we look at this homologous chromosome here, you will see that these homologous chromosomes here, one coming from the parent, it has got a gene at this location. And also on the same location on the other chromosome, there is also a gene. Now, those genes at those particular points, they control the same characteristic. Like for example, this one controls the eye color. And also this one, you will see it also a gene which controls the eye color. And still, if we go to this other homologous chromosome here, you are going to see that at this particular point here and this particular point here, we have a gene. And this gene here is controlling the type of hair. And this one also, it's a gene which is controlling the type of hair. So that is what we are saying, that the genes for any one characteristic occupy a corresponding position on a homologous chromosome. So at this point, we are having a gene that is controlling the eye color. And when you come to this one, at this position here, we are having a gene which is controlling the type of hair. So we can go to other points. You may find that here we are having another gene, and also here we shall find another gene. These genes will be controlling the same characteristic at that corresponding position. But they do not necessarily control the characteristic in the same way. It's not that because they are controlling the same character, they will be controlling it in the same way. No, it may not be necessarily that way. For example, here when you look, it is a gene for brown eyes, whereas here it is a gene for blue eye. They are different. Sometimes they may be the same, but another time they may be different. For example, here it is a gene for curl hair, and here it is a gene for straight hair. For example, one of the gene pair corresponds for eye color might determine the brown eyes, and its pattern determines for blue eyes. Like for example, here we are having a gene for brown eyes, and the other side here we are having a gene for blue eyes. So that's what we are saying, that they may not be necessarily controlling the eyes in the same way. And the eye color and the hair colorness are not controlled by a single gene. The eye color and the hair colorness, they are not controlled by a single gene. For example, here we are looking at eye color, and you are seeing that it is not a single gene which is controlling the eye color. This is not only a single gene, it is controlled by more genes. And here you are seeing we are having the hair colorness. It is not controlled by one gene, but it is controlled by very many genes. A person inheriting the gene for brown eyes and the gene for blue eyes will have brown eyes. For example, if you look here, this person inherited a gene for brown eyes, and he also inherited a gene for blue eyes. Now, which type of eyes is this person going to have? Because he has both the genes, one for blue eyes and another one for brown eyes. But here we are having the person becoming with brown eyes. The gene for brown eyes is said to be dominant to the gene for the blue eyes. So if this person inherited the gene for brown eye, which came from the male parent, and he inherited the gene for blue eyes, which came from the from the mother's side, this person is ending up with the blue eyes. Why? Because the gene for the blue eyes is dominant to the gene for the brown eyes is dominant to the gene for the blue eyes. Therefore, this person is going to become brown-eyed because the gene for blue eye is dominant over the gene for brown eyes is dominant to the gene for the blue eyes. The gene for the blue eyes is not expressed in this individual. So you are finding that this gene for the blue eye is not going to express itself because it is being dominated by the gene of the brown eye. It does not mean that it is not existing. It is existing, but it is going to be suppressed. It will not be allowed to express itself. The gene for the blue eyes is said to be recessive to the gene for the brown eye. So our gene for the blue eye is recessive, meaning that it is dominated by the gene of the brown eye. Therefore, we call it a recessive gene. The gene for the blue eye is now called a recessive gene because it is being dominated by the gene of the brown eyes. The gene, for example here, when we are having our cell division, this cell up here is taking the gene for straight hair. And this one has taken the gene for the curl hair. It is taking the gene for the curl hair up, and this one down has taken a gene for the straight hair. Then the cell down, this cell down here, this cell down, as we have seen, it has taken a gene for straight hair, and it is also taking a gene for blue eyes. So it has taken also a gene for the blue eyes. So if it takes a gene for blue eyes, at the same time it has also taken a gene for straight hair. So when you look at this person down here, the person down has taken a gene B for blue eyes and a gene C for straight hair. Then the other person up has taken a gene for brown eyes. So now when we look at these two cells, the cell up has taken a gene for the brown eyes, and it has also taken a gene for curl hair. Then the one down, the cell down, has taken a gene for the blue eyes, and it has also taken a gene for the straight hair. So the gene B, small b, and capital B are said to control a contrasting character, either blue or brown eyes. So when you look at this one here, this is capital B, and this one is small b. They are all controlling eye colors, but one has got brown hair, brown eyes, and another one has got blue eyes. So they are contrasting characters. It is the same character but contrasting each other. One is blue-eyed, and then another one is brown-eyed. So they are all eyes, but they are contrasting. This one is blue eye, and this one is brown eye. So they are contrasting character. Similarly, curl hair and straight hair are contrasting character. So you can see that up here we have the curl hair, and this one is for straight hair. All of them, they are controlling hair, but they are contrasting each other. So they are contrasting characters. So one gamete will receive gene combination for brown eyes and curl hair. So we have one of them is going to receive brown eyes and curl hair. Then the other will receive genes for blue eyes and straight hair. The other one will receive, these are blue eyes, and straight hair. And then the upper one we have brown eyes and curl hair. So the cell up is going to have brown eyes and curl hair. And then the one down will have blue eyes and straight hair. Now, one gamete will receive the gene B, capital B, and C, small c, which is brown eyes and straight hair. So we are seeing that this cell up has received the genes for the brown eyes and the straight hair. If you look at it, it has got brown eye and straight hair. So this gamete is going to have brown eye and straight hair. Then the other gamete will receive gene small b and capital C, which are the genes for blue eyes and curl hair. So we are having now the lower gamete is going to have a gene for blue eyes and a gene for curl hair. So those gametes are going to have contrasting characters. In terms of eyes, this one will have brown eyes and this one will have blue eyes. Then when it comes to hair, this one will have straight hair and this one will be having curl hair. So there are 4 types of gametes with different combinations of genes. So we are going to get 4 types of gametes, but with different combinations of genes. We shall have BC, B capital C. This one, the gamete up, can have B, which is capital, and C, which is capital, meaning that it has got brown eyes and curl hair. Then we are going also to receive smaller b, smaller c, and you will see that this one has blue eyes and straight hair. So the gene that gamete we are having at the lower side here is going to have the blue eyes, the gene for blue eyes, and the gene for straight hair. Then there are other possibilities. We can have the capital B and the smaller c. So we can have another cell, and this cell may be of this type, where we are having capital B and a small b. For example, we may have another cell which may take this chromosome and this chromosome. So it will be now having capital B and then smaller b, which are genes for brown eyes and straight hair. There is that possibility that can happen also. Then we can have another possibility where we have smaller b, gene smaller b, and a capital C. So here we can likely to have a gamete which can take that gene, and it also takes this gene here. It can take those genes, meaning that it has a gene for smaller b, which is blue eyes, and it has a gene for capital B, which is curl hair. So this gamete will now have blue eyes, and then it will also have curl hair. So those are the possibility of combinations of genes. So we can end up having 4 gametes with different combination of genes. We have seen we can have capital B and a capital C. This one is a gamete which has brown eyes and a curled hair. Then we saw that we can also have small b and small c. This gamete will be having blue eyes and a straight hair. Then we have seen that we can have a gene or a gamete with capital B and a smaller b, meaning that this gamete will be having a gene for brown eyes and a straight hair. And we have also seen that we can have a gamete taking small b and capital C, meaning that this gamete will be having a gene for blue eyes and curl hair. Meiosis not only halves the number of chromosomes, but also rearranges the genes. It rearranges. We have seen that meiosis leads to reducing the number of the gametes to a half, but also rearranging the chromosomes or the rearrangement of the genes in the different gametes. This is one cause of variations that occur in members of the same species. That meiosis, whereby there is a rearrangement of genes, is going to bring about variation in the members of the same species. For example, if you look at these cats, you will see that there is variation. This is maybe the mother. If you look at the mother, you can look at the colors of the mother, and then you also look at the colors of the kids. kittens. You can see that the kitten, they have variation in their colors. This one, you look at its color, is different from this one. This one tries to resemble that one of the mother, but you can see that this one has got a variation. That is brought about by the rearrangement of genes, which is brought about by meiosis, which brings about variation in the members of the same species. Rearrangement of the genes can also take place at fertilization. Rearrangement of genes can also take place at fertilization. For example, let's look at these 4 cells. We have, this is a sperm with capital B, but we also have a sperm with small b. And then we have the ovum. The ovum up, we have the ovum with capital B, and then we have another ovum with smaller b. Now, when you look at these gametes here, if you look at these gametes here, let us try to see them here. We can give this one a different color so that we can try to differentiate them. Now, when you look at this one, let us say this sperm here goes with this ovum here. We are going to get capital B and capital B. So here we are going to have this one here, capital B, and then the other capital B, the other side. Okay, so that is going to be our combination there. So now again, if we say, let this sperm go now with this ovum here. So this is capital B, this is small b, which means it is going to produce this one here. It is going to produce this one, small b and capital B. So what we are going to get here, we are going to have our small b, and then the other side we are having the capital B. So that is now the offspring, the combination we are getting here. Now, if we move on from that one and then we go to this sperm here, let us say it is now this sperm which is taking part in fertilization. It may go with this ovum here. When it goes with this ovum here, which is capital B and this is smaller b, we are going now to get here capital B, smaller b. So here we are going to get a combination of capital B, and then we shall get the smaller b this side. That is going to be our combination there. Now, if we'll say that this sperm is going to go with this ovum here, both of them are small b, smaller b. So here we are going to get our smaller b, and then we shall also get the other smaller b. So if you look at the combination, those are the 4 possible combinations. When you look at them, here yellow has gone with black, here black has gone with red, here blue has gone with yellow, red has gone with blue. So you can see that there is going to be variation in the offspring. This offspring will not be looking the same as this one. Will not be looking at the same as this one. There is going to be some variation genetically. So the sperm, our sperm may carry the gene for brown eye, which is capital B, or it may carry the gene for blue eye. So the sperm here is carrying a gene for the brown eye. At the same time, it is carrying a gene for blue eye here. Then the ovum may carry a gene for brown eye and also carry a gene for blue eye. So when you look at our ovum, here it is carrying a gene for brown eye, and here it is carrying a gene for blue eye. So after fertilization, 4 possible combinations are going to occur. We are going to get 4 possible combinations. Capital B, capital B, capital B, smaller b, capital B, and smaller b, smaller b. Those are the 4 possible combinations that we are going to get. Although there are 4 possible combinations of genes, capital B, capital B, capital B, smaller b, capital B, these ones have the same effect and producing brown eyes. So we will see that this one will produce brown eyes, this one will produce brown eyes, and this one will produce brown eyes. Why? Because there is this dominant gene. It's going to suppress this one. So this one will be brown eyes. This one is a dominant gene, will suppress this one, and this one will be brown eyes. And these two, both of them are dominant genes, so this one will be brown eye. So you will find that all of these combination, they are going to have brown eye, even if they have some recessive gene, but this recessive gene is going to be suppressed by the dominant gene. Only small b will give the blue eyes. So this one will only be the one having blue eyes. Why? Because it is small b and smaller b. Both of them are recessive, and therefore both of them will express themselves, and the offspring will have the blue eyes. Thank you very much for participating in Chromosomes and Fertilization. Please do like and share.